You know that glycolysis results in pyruvate. Based on the availability of oxygen, pyruvate either undergoes anaerobic or aerobic respiration. Aerobic respiration involves the transport of pyruvate from cytoplasm into the mitochondria for complete oxidation in Krebs cycle. It also involves the transfer of hydrogen ions and electrons from the coenzymes NADH plus H plus and FADH2 to oxygen in the electron transport system, or ETS, for ATP synthesis. Now, let's see how the complete oxidation of pyruvate takes place. Inside the mitochondria, the pyruvate undergoes oxidative decarboxylation in the presence of the pyruvate dehydrogenase enzyme NAD+ and coenzyme A. The NAD+ is reduced to NADH plus H+, and coenzyme A combines with the acetyl group of pyruvate to form acetyl CoA. The acetyl coenzyme A thus formed enters Krebs cycle, which has been named after the scientist Sir Hans Adolf Krebs. Krebs cycle starts with the reaction of acetyl coenzyme A with oxaloacetic acid to form citric acid, catalyzed by the enzyme citrate synthase, with the release of coenzyme A. The first product of Krebs cycle is citric acid, and hence the cycle is also called the citric acid cycle or the tricarboxylic acid cycle. The citric acid thus formed isomerizes to form isocitric acid. The isocitric acid is then oxidized in the presence of NAD+ to form oxalosuccinic acid, which is later decarboxylated to form alpha-ketoglutaric acid. This alpha-ketoglutaric acid undergoes oxidative decarboxylation using coenzyme A to form succinyl coenzyme A, along with the reduction of NAD+ to NADH plus H plus ion. The succinyl coenzyme A formed is hydrolyzed to succinic acid with the release of coenzyme A. This reaction also results in the production of guanosine triphosphate or GTP, a high-energy molecule from GDP, by substrate-level phosphorylation. The GTP thus formed is again converted back into GDP, coupled with the synthesis of ATP from ADP. The succinic acid formed in the previous reaction is oxidized to fumaric acid with the reduction of FAD+ to FADH2. The fumaric acid is hydrolyzed to malic acid, which is then oxidized to oxaloacetic acid with the reduction of NAD+ to NADH plus H plus ion. For Krebs cycle to run continuously, oxaloacetic acid, NAD+, and FAD+ need to be regenerated. The summary equation of Krebs cycle is as shown here. The net gain of Krebs cycle in the conversion of 2 molecules of pyruvate is 2 molecules of ATP, 8 molecules of NADH plus H plus, and 2 molecules of FADH2. So far, the energy harvested has resulted in the direct synthesis of only 4 molecules of ATP. Further generation of energy as part of aerobic respiration takes place in the electron transport system or ETS.